Hey everybody, today we're going to be talking about regulation of gene expression. We have already discussed the central dogma of molecular biology. This is the process whereby we use our DNA blueprint to make RNA, and then we use the RNA to make proteins. We're going from the language of DNA and RNA, which are nucleotides, to the language of proteins, which are amino acids. When we go from DNA to RNA, we call that process transcription. The star of the show for transcription is RNA polymerase. When we go from messenger RNA to making amino acids, that's called translation. And we use a ribosome along with tRNAs to accomplish that process. When you go through this entire pathway using DNA as a blueprint to make messenger RNA to ultimately make a protein, it's called gene expression. Energy is precious to the cell. We have already learned all about the stuff the cell needs to do to make ATP. The cell doesn't want to use up its ATP unless it has to, because that would be wasteful. The cell has to regulate what it makes and when it makes it. There are two main mechanisms that the cell uses to regulate transcription. The first is feedback inhibition, and the second is gene regulation. We have already talked about feedback inhibition. You can see here that we have a metabolic pathway that makes the amino acid tryptophan. The precursor molecule is at the top, and we need three different enzymes to help catalyze the reactions to make tryptophan. In feedback inhibition, accumulation of the end product can shut down the pathway because the end product, in this case tryptophan, can inhibit an enzyme earlier in the pathway. Feedback inhibition is fairly short term and adapts quickly to changing conditions in the cell. On the right side, you can see an example of repression of expression. This is also based on the amount of the end product. However, the regulation of expression is a longer-term solution to reduce waste. In this case, the end product, which is tryptophan, is going to inhibit transcription of the enzymes themselves. If there's no transcription of the enzymes, there'll be no translation of the enzymes, there'll be no actual enzymes there. This leads to the cell wasting less resources in two ways. Not only is it not producing excess tryptophan, but it's also not producing any of the enzymes in the pathway to make tryptophan. So it's a double savings. This mechanism takes longer to turn on and off than feedback inhibition, and it involves changing the amount of the enzymes themselves, not just inhibiting one of them from working. To turn on and off genes, prokaryotes use something called an operon. This serves as a switch to turn several related genes on or off at the same time. Think of the lights in your house. When you leave to go out, you have two choices. You can either turn every lamp in your house off individually, or you can go to the fuse box and turn off all the power to the house in one step. A group of genes that are related and can be controlled as a group is called an operon. The part that serves as the switch is called the operator. This is a section of DNA within the promoter region. A quick review of transcription. Here's some DNA. You can see a group of genes. They all do a similar function and they're clustered together. The RNA polymerase will bind to the promoter of the DNA upstream of the genes. RNA polymerase will move to the right towards the genes until it comes upon the initiation sequence. This is the signal to start transcribing the genes into messenger RNA. Now here's the new information. There is a site between the promoter and the genes called the operator. This site can either turn on or turn off transcription. The entire stretch, including the promoter, the operator, and its related genes, is called the operon. There are two types of operons. A repressible operon is one which is normally on, but you can use the operator to switch it off. An inducible one is normally off, but you can use the operator to switch it on. Let's talk about the lac operon. This is an inducible operon. The lac genes in the lac operon code for enzymes to break down lactose. When there is no lactose in the cell, You don't want to waste resources making all those enzymes. It's wasteful. So the lac operon is inducible. Normally, it's off. In an inducible operon, a repressor protein binds to the operator site. This creates a roadblock where RNA polymerase cannot get by to begin transcription. No transcription, no translation, no protein, no enzymes. However, if there is lactose in the environment, the lac operon can be turned on or induced. If there is an inducer, it will bind to the repressor and cause it to change its shape. It can no longer bind to the operator now, and the inactivated repressor falls off the DNA. The roadblock has been removed, and transcription of the genes will begin. Another operon example is the trip operon. The genes that are regulated here make tryptophan. This is a repressible operator, which means it's normally on because the cell normally needs a lot of tryptophan. This operon can be repressed or turned off if you don't need as much tryptophan. You can see here that there's nothing blocking the operator site. RNA polymerase can bind to the promoter and start transcription at any time. There is a repressor, but it's inactive. It cannot bind to the DNA because it's in the wrong shape. However, when tryptophan levels in the cell start to build up, for example, if the cell stops growing, the tryptophan can bind to the repressor protein. This causes the repressor to change shape, and now it can bind to the operator. This blocks RNA polymerase from starting transcription. This is similar to feedback inhibition and is based on the level of tryptophan in the environment. The big difference here is that gene regulation will reduce the amount of enzymes produced to turn off a pathway. This regulation is generally reversible. When tryptophan levels fall back down again, it will no longer bind to the repressor, and the repressor will go back to being inactive, allowing transcription to proceed and more end product to be made. Inducible operons usually function in catabolic pathways. Their synthesis is induced by a chemical signal. Repressible enzymes, however, usually function in anabolic pathways. They are repressed by high levels of the end product. We know that all cells in our bodies have the same genome, but different types of cell, say your liver cells versus your neurons, have different proteomes. The amount and type of proteins in different cells is different. Have you ever wondered how cells do this? In order for cells to have specialized functions, each cell must regulate which genes and how much of each gene is expressed. This is called differential gene expression. Gene expression can be regulated in many ways. It can be regulated at the level of DNA, RNA, or proteins. Regulating gene expression at the level of DNA involves whether or not the DNA is accessible. Recall that chromatin is DNA plus histone proteins all wrapped up around each other. When you have 8 histones wrapped together, it forms a little ball called a nucleosome. Think about the structure of the nucleosome. The DNA on the outside of the little ball is much more accessible than the DNA segments that are wrapped inside the ball. You are more likely to be able to transcribe the DNA on the outside of the nucleosome, or maybe between nucleosomes, than the DNA that's stuck on the inside of a nucleosome. This leads to differential gene expression. The DNA that is very tightly wound is called heterochromatin. The DNA that is looser and accessible is called euchromatin. There are also some chemical modifications that can be done to the histones to encourage or discourage gene expression. Adding acetyl groups to histone protein makes the DNA looser, more available, and therefore more likely to be expressed. Adding methyl groups to the histones condenses the chromatin, allows it to coil much more tightly, which makes it harder to access. In this way, chemical modification of the histone proteins can change the expression of your DNA. These modifications are called epigenetics or epigenetic inheritance. These are hereditable, but can sometimes be reversed, unlike mutations to the DNA. This is why one identical twin can get a genetic disease, such as schizophrenia, while the other twin doesn't get it. The change is not in the actual DNA sequence itself, but in the structure of the chromatin. We can also change gene expression by regulation at the level of transcription. Messenger RNA has a lifespan. How many times can you translate each transcript before it gets degraded? This is going to influence how much protein you can make. You can stabilize the RNA if you want it to last longer. Another way you can regulate gene expression at the level of transcription is to splice the RNA in alternate forms to give different gene products. That's shown here. The exons are the part of the genetic material that will eventually make it to the messenger RNA after processing. The introns are removed. Sometimes you can splice the RNA in different ways so that, for example, on the left, you have exons 1, 2, 3, and 5, but on the right, that gene product is going to be 1, 2, 4, and 5. This is going to give you two very different proteins from the same exact DNA. Another way of regulating gene expression is during translation or after translation. The initiation of translation of selected messenger RNAs can be blocked by regulatory proteins that bind to sequences or structure of the mRNA. It will block translation at the level of the ribosome. Here's an overview of the different ways that gene expression can be modified. The blue box contains modifications that can be made in the nucleus to the DNA. The orange boxes are modifications that can be made before translation, and the purple boxes are changes that can be made during or after translation. Cells can use a combination of all of these options to upregulate or downregulate expression of various genes. This allows a multicellular organism to make highly organized tissues that consist of different types of cells. These cells have different functions, even though all of the cells in an organism have the same genetic material. That's it for today. See you in class.